In this nervous system part 6, we will cover reflex arc, neurotransmitters, neural tracts, brain waves, sleep and sleep patterns, circle of Willis. A reflex action is a rapid involuntary response to a stimulus, and many have a protective or survival function. Examples of reflex actions are pupil reflex, reducing the size of the pupil in bright light to protect the retina. Coughing. Coughing prevents asphyxiation or suffocation, which occurs when the body is deprived of oxygen. Blinking reflex protects the eyes by keeping the surface moist. Withdrawal reflex, for example, jerking the hands away when touching a hot surface. Reflex actions follows a specific nerve pathways known as reflex arc. Reflex arc involves the following anatomy: the receptor, the dendrite of a neuron receiving the stimulus. The example of the stimulus is a chemical, heat, light, or mechanical disturbances. Next is afferent neuron or sensory neuron, a neuron that has an action potential carrying the signal to the CNS. Sensory receptors detect the stimulus and convert it into impulses transmitted along sensory afferent neurons to the CNS. Integrating center, either the brain or spinal cord, where the signal is received from the afferent neuron and conducted to a motor neuron. This may or may not recur an interneuron. Efferent neuron, a neuron that has an action potential carrying a signal away from the CNS. Motor neurons transmit the impulses along efferent neurons to a muscle or gland, which responds by producing the right reaction. Effector, the structure causing the effect. If this structure is skeletal muscle, it is called a somatic reflex. If the effector is a gland or smooth muscle, it is called an autonomic reflex. Synapse. A synapse is a junction between any two communicating neurons at which the nerve impulses passes from one to another. The actual gap between the neurons is known as the synaptic cleft. Neurons conduct intracellular communication across these gaps. The nervous system requires impulse transmission through neuron chains that are functionally connected by synapses. A neuron carrying an impulse into a synapse is called a presynaptic neuron. The neuron receiving this impulse is called a postsynaptic neuron. The process of the impulse crossing the synaptic cleft is called synaptic transmission. Most neurons function as both presynaptic and postsynaptic neurons. Synaptic transmission occurs in one direction carried by biochemical, that is, neurotransmitters. The axon of the presynaptic neuron breaks up into minute branches that terminate in small swellings called synaptic knobs or terminal buttons. Synaptic knobs contain spherical synaptic vesicles, which store a chemical, the neurotransmitter, that is released into the synaptic cleft. What is neurotransmitters? A neurotransmitter is a signal molecule, a chemical messenger that is released from a presynaptic neuron and diffuses across the synaptic cleft to either excite or inhibit the target cell in order for communication to occur between neurons or between the neuron and muscle or gland. Those effector cells or neurons in close proximity to the neurotransmitter will either be stimulated or inhibited by the neurotransmitter, depending upon which neurotransmitter is secreted. The action of the neurotransmitter is short-lived, and any neurotransmitter not used is absorbed by the neuron to be recycled and used again or deactivated by enzymes. Some examples of neurotransmitters are acetylcholine, released within the CNS and also at the neuromuscular junction. Norepinephrine, released within the central nervous system and also at autonomic nervous system synapses. Dopamine, released within the central nervous system and also at autonomic nervous system synapses. Neurotransmitters have special characteristics. They are made in the neuron and stored in vesicles in the axon terminal on the presynaptic side of a synapse. When the neuron is stimulated, they are released and produce a specific response in the neighboring target cell, that is postsynaptic cell. They are then degraded and removed or taken back into the axon terminal for reuse. Over 100 neurotransmitters have been identified and they play a major role in many of the body's activities, for example, mood, sleep, appetite, concentration, or memory. It works best when present at their optimum level, and adverse symptoms may result when they are out of balance. It can be depleted in many ways, for example, by stress, poor diet, genetic predisposition, drugs, and neurotoxins. Most neurotransmitters are either excitatory or inhibitory. Excitatory neurotransmitters increase the likelihood that the target cell will produce an action potential and transmit an impulse, for example, acetylcholine. Inhibitory neurotransmitters act by reducing the membrane potential, so it is further away from threshold, making an action potential less likely, for example, gamma aminobutyric acid. Next is neural tracts. Within the white matter of the spinal cord are bundles of axons called tracts that serve as the routes of communication to and from the brain. All the nerve fibers of a single tract have a similar origination, destination, and function. As an example, the fibers of a spinothalamic tract originate in the spinal cord, that is spino, and end in the thalamus, thalamic. In addition, they all convey sensations of pain, touch, and temperature to the thalamus in the brain. Neural tracts and pathways connect distant parts of the nervous system with each other by means of bundles of nerve fibers in the white matter. Ascending tracts are bundles of afferent nerve fibers that carry sensory impulses from the peripheral nerves to brain. For example, relating to touch, heat, and pain. The thalamus acts as a relay center, directing sensory impulses to the appropriate part of the cerebral cortex for processing. Descending tracts are bundles of efferent nerve fibers that carry impulses from the motor cortex via the spinal cord to control movements of the body below the head. The reticular activating system comprises bundles of neurons in the brainstem that are responsible for sleep, wakefulness, and level of alertness. The corpus callosum is a bundle of nerve fibers that cross the midline of the cerebrum, enabling the transfer of information between the two cerebral hemispheres. Next is brain waves. Brain waves are based on electrical activity, and normal brain functions involve continuous electrical activity of the neurons. Certain aspects of electrical brain activity can be recorded on an electroencephalogram, which involves placing electrodes on the patient's scalp. The EEG measures voltage differences between the areas of the cerebral cortex. Brain waves are the patterns of neuronal electrical activity that is recorded. They are generated by the activity of the synapses at the surface of the cortex. Every individual's brain wave patterns are unique, but are grouped into 4 primary types. First is alpha waves. Relatively regular, rhythmic, synchronous waves of low amplitude, that is 8 to 13 Hz. They usually indicate calm and relaxes wakefulness. Beta waves, rhythmic but less regular waves that have a higher frequency than alpha waves, 14 to 30 Hz. They occur during mental alertness, such as when concentrating or looking at visual stimuli. Next is theta waves, irregular waves that are common in children and have a low frequency, that is 4 to 7 Hz. They may occur in adults when concentrating. Delta waves is high amplitude waves occurring in deep sleep or when something dampens the reticular activating system, that is such as anesthesia. If These waves exist in a conscious patient. They indicate brain damage. Brain waves changes with brain diseases, aging, sensory stimuli, and the chemical balance of the body. Next is sleep and sleep patterns. Sleep is a state of partial unconsciousness from which we may be aroused by stimulation. It is different from coma, from which a person cannot be aroused by stimulation. If the person is sleeping, we can arouse them, but if the person is in coma, we cannot arouse them. During sleep, most cortical activity, that cerebral cortex, is depressed, but brainstem functions continue. These functions include control of heart rate, blood pressure, and respiration. The two major types of sleep are non-rapid eye movement sleep and rapid eye movement sleep. Each of these have different patterns on an EEG. Stages of sleep. Stage 1 is non-rapid eye movement. Relaxation begins in this. Arousal is very easy. In this, EEG shows alpha waves. The second one is non-rapid eye movement 2. Arousal is very difficult. EEG is irregular with the sleep spindles, that is short, high amplitude bursts. In third stage, sleep deepens and vital signs decline. On EEG, theta and delta waves appear. In fourth stage, arousal is very difficult. Bedwetting, nightmares, night terrors, or sleepwalking may occur. EEG is dominated by delta waves. The transition to REM sleep from this stage is marked by an abrupt change in EEG patterns, with alpha waves reappearing. In rapid eye movement sleep, most dreaming occurs. Except for diaphragm and ocular muscles, the skeletal muscles are actively inhibited, which prevents acting out of dreams. More oxygen is used during REM sleep than during consciousness. Next, we will see about circle of Willis. The circle of Willis, circulus arteriosus cerebrae, is an anastomotic system of arteries that sits at the base of the brain. The brain lies in the cranial cavity and weighs between 1450 gram and 1600 gram. It receives 15% of the cardiac output and has a system of auto-regulation ensuring the blood supply is constant despite positional changes. The arrangement of the arteries serving the brain is unique and they are connected to each other by structure called the circle of Willis. This arrangement ensures that blood pressure remains equal in both halves of the brain. If one of the arteries serving the brain become narrower by arterial disease or thrombus, then there will be an alternative route available, maintaining the essential supply of oxygen and glucose required by the brain. The circle of Willis encircles the stalk of the pituitary gland and provides important communications between the blood supply of the forebrain and hindbrain. The circle of Willis is formed when the internal carotid artery enters the cranial cavity bilaterally and divides into anterior cerebral artery and middle cerebral artery. The anterior cerebral arteries are then united by an anterior communicating artery. These connections form the anterior half of the circle of Willis. Posteriorly, the basilar artery formed by the left and right vertebral arteries branches into a left and right posterior cerebral artery, forming the posterior circulation. The posterior cerebral arteries complete the circle of Willis by joining the internal carotid system anteriorly via the posterior communicating arteries. Although in an adult the brain represents only 2% of the total body weight, it utilizes 20% of oxygen and glucose even at rest. When activities in a certain area of the brain increases, blood flow to that region also increases. Even a brief slowing of blood flow to the brain can result in unconsciousness. Further, decrease in blood flow for a couple of minutes can lead to impaired neuronal function. If the blood flow is restricted for 4 minutes or more, then it could lead to permanent brain damage. As the brain does not store glucose, there must be a continuous supply of glucose to the brain. What are the functions of the circle of Willis? The circle of Willis provides multiple paths for oxygenated blood to supply the brain if any of the principal suppliers of oxygenated blood are constricted by physical pressure, occluded by disease, or interrupted by injury. This redundancy of blood supply is generally termed collateral circulation.